Welcome back. In this section, we're going to talk about eukaryotes. So we're on Unit 7. The first video I made talked about the prokaryotic organisms. So now we're going to talk about characterization and classification of eukaryotes. Now, the eukaryote microbes are going to include species that are going to be both vital for life and human pathogens. So we have 6 known species that can cause death worldwide. The major groups of eukaryotes are going to include the protozoa, fungi, algae, water molds, and slime molds. In addition, microbiologists are also going to study things like parasitic helminths due to their microscopic stages and arthropod vectors because of their involvement in microbial pathogen transmission. Now, starting us off, we're going to discuss eukaryotic reproduction. So there's going to be 2 divisions of eukaryotic reproduction. We're going to have the nuclear division, and there's going to be 2 types of that even. So within these 2 types, the first one we're going to talk about is mitosis, and that's what you see here on the screen. That's going to result in 2 nuclei with the same ploidy as original. So haploid is going to be a single copy of each chromosome. So cells are going to form 2 haploid cells. Diploid is going to be 2 sets of a chromosome, so the cell's going to form 2 diploid cells. And so with this, basically, you're going to take a cell, it's going to double up its genetic information, so it goes, let's say if it's human, it would go from 46 chromosomes to 92. Then it would go through a division to make 2 identical cells in the end. And so with this, you're going to have 5 main stages. You have interphase, which is going to be cell growth. That's where a cell spends most of its time. Here it is going to go through what's known as G1, S phase, and G2. G1, or growth phase, is where the cell grows and does its job. S phase is going to be where you have DNA replication, and then it's going to prepare in G2 for the cellular division or nuclear division. Nuclear division is going to begin with prophase, and that's what you're going to see here in the second picture. So we're going to start out with prophase, and an easy way to remember this, a lot of people remember it as PMAT. So during prophase, your DNA is going to condense into chromatids, and the spindle apparatus is going to start to form. And so that's going to start to come out of these centrioles that are making their ways towards the poles, so each end of the cell. Metaphase, you're going to have those chromosomes lining up on the plane right directly through the middle of the cell. Think of it like the equator. Notice how you have your centrioles now at each end of the cell, and you're going to see those spindle fibers starting to come out of those centrioles. Now they'll come down and they're going to attach to the centromeres of these chromosomes. And so when they line up, you see those microtubules coming out of the centromeres. They come in and then they attach at the centromere. Anaphase is going to be this next phase that we see coming in, and that's where the sister chromatids are going to separate and move to the opposite poles of the spindle from that chromosome. So you see those those centromeres are going to start to retract, or those spindle fibers, those microtubules, are starting to get shorter. So they're attached at these centromeres, these little dots that you see on each of the chromatids, and so they start to pull them to one pole or the other pole. Also during this time, you start to see some pinching in here. That's going to be called cleavage. So that's a cleavage furrow. Telophase is where that nuclear envelope is going to start to reform around those daughter nuclei. So you see a nucleus start to reform. It's definitely pinched in a lot here, and it's starting to pull apart. When it pulls apart, that is going to be referred to as cytokinesis. And then you went from one daughter cell here, and one parent cell, and you're going to end up with 2 daughter cells here. The second type of asexual reproduction in eukaryotic cells is going to be meiosis. It looks quite similar to mitosis, but it's going to actually go through those phases twice. So with meiosis, like we said, it occurs in 2 stages. We're going to have meiosis 1 and meiosis 2. First thing is going to double up those chromosomes in the S phase before it actually enters into meiosis. And so it goes from the 46 normal chromosomes, let's say of a human, to 92. So from there, we want to divide it in half. So by the time that we get to the end of the first meiosis here, we have 46 chromosomes instead of the 92 that we started with. And then by the time you get here, you want to end up with 23 chromosomes in each one of those cells. So this is going to be for gametes in particular, so for sex cells. So with this, our 4 main stages, we start out with prophase 1. Early prophase, we're going to have the formation and lining up of the homologous chromosomes. Chromosomes are going to carry identical genetic sequences, and they're lined up in what's called a tetrad. They're aligned in pairs of homologous chromosomes. By late prophase, you're going to have crossing over occur. That's where if one of these chromosomes gets too close to each other, they're actually going to switch ends of their legs. So let's say a little piece of the red might go to the green and some of the green might go to the red. So that would be called crossing over. Only occurs in prophase 1. Next, we're going to go to metaphase 1. That's where the tetrads are going to align in the center of the cell and attach to the spindle microtubules. Anaphase 1 is where the tetrad chromosomes are going to start to move apart. Here, your sister chromatids are going to remain attached. Telophase 1, that spindle's going to start to disintegrate. Cell's going to start to divide to form a haploid nucleated daughter cell. And then it sits for a short period, kind of like a little interphase, and then goes into meiosis 2. Here, it's going to have the same 4 main stages, but we're going to put 2 behind it. So now we're going from the 46 chromosomes starting out here, and hopefully we'll end up with 23 by the end. So in prophase 2, you see that nuclear envelopes start to disintegrate with new spindle forming. Metaphase 2, you have those chromosomes aligning in the middle of the cell and attaching to the spindle microtubules. By anaphase 2, the sister chromatids separate, and by telophase 2, daughter nuclei are going to form and cells divide, producing 4 haploid cells. So these up here are 2 diploid cells, these right here, 2 haploid cells. Once you divide the nucleus, which is mitosis or meiosis, then you, of course, are going to divide that cell. So once you divide that cell, that is referred to as cytokinesis. This is cytoplasmic division. So it occurs in conjugation with mitotic telophase. Schizogony is going to be asexual reproduction in which multiple mitoses or mitoses are going to occur, forming many daughter cells. It produces multinucleated schizone. This occurs in some protozoan species like the Plasmodium. The classification of eukaryotic microbes is going to be problematic, and it's going to have a lot of change, so it's going to change quite frequently. Historical schemes are going to be based on similarity in morphology and chemistry, and they've been replaced with schemes that are going to be based on DNA and RNA, or what we refer to as nucleotide sequences and ultra-structural features. So with this, we're going to have our 5 main microbial eukaryotic groups. We're going to discuss the protozoans, the fungi, algae, water molds, and helminths and vectors. So starting out with our protozoans. The protozoans' main characteristics are that they are eukaryotic, they're unicellular, which means they're made up of one cell, they lack a cell wall, and then they can be compared according to their distribution. So with protozoan distribution, some are going to be colonial, which means they make colonies, and then some are going to form loose aggregations. Most are going to live and function as separate cellular individuals. They require moist environments like ponds, streams, lakes, and oceans. They often live in things like moist soil, beach sand, or decaying matter, and they can be pathogens of animals and of humans. Now with protozoan morphology, their locomotion is going to be cilia for some, and some are going to have flagella, or some may even use pseudopodia. That's where they have extensions of their cytoplasm. Some are going to have 2 nuclei, and some are going to lack mitochondria. Some are going to have contractile vacuoles. They're going to function as osmoregularity structures, so they prevent the cell from bursting due to osmosis or bringing in too much water. They're going to aid in fluid circulation within the cell, and they may also have an extra excretory function where they can excrete if there's too much waste or too much water in it, they can get rid of that. We have the trophocyte, which is going to be a motile feeding stage, and you can find it as a cyst. And so cysts are going to be hardy resting stages that are characterized by thick capsules, and they're going to have a low metabolic rate. Now, protozoan nutrition. These are going to be chemoheterotrophic, which means they rely on chemicals from other things. So using preferred organic compounds for both energy and carbon, they're going to obtain these from phagocytizing bacteria, from decaying organic matter, from other protozoa, or from the tissue of the host that they're living on. Few protozoa are going to absorb nutrients from the surrounding water that they may be in. Now, protozoan reproduction with asexual, we have two types. We have asexual and sexual. So two types. As far as the asexual, we're going to reproduction, that's going to be without the interaction of other cells. So with the, you'll have the binary fission, schizogamy, and budding. So the binary fission, you're going to have the parental cell dividing into two identical daughter cells, and those parental cells are going to disappear with the formation of their offspring. With schizogamy, you're going to have a reproduction in which multiple mitosis occurs, and that's going to form a lot of daughter cells. With budding, that's going to be the process which outgrowth of the parent cell is going to receive a copy of genetic material, enlarge, and then detach from the parent cell. With sexual reproduction, there's going to be two main types. You have syngamy, that's going to involve the fusion of two haploid cells to form a diploid zygote. Plasmodium are going to undergo syngamy while inside a mosquito. And conjugation. Conjugation is going to require that two cells with different genetic content meet side by side. Cell membranes are going to then fuse, allowing the formation of cytoplasmic bridges. You're going to have segments of genetic material or DNA that are going to be exchanged between the two cells. Following this, the cells will then separate. Now we need to go into protozoan classifications. So one of these classifications could be a parabasala. That's going to lack mitochondria, and it's going to contain a single nucleus. The parabasal body is going to be a Golgi-like body or structure, and it receives, processes, and packages large molecules in secretory vesicles that are going to be released through exocytosis. So here in the picture you see Trichomonas. This is going to live within the female vagina. This is going to cause trichomoniasis. That results in inflammation and a creamy white discharge with severe itching and chafing. Transmission is going to be through direct sexual contact, and it's generally going to be asymptomatic in males, but could infect the urethra and the prostate. With our next one, we have Diplomonadia. In this one, it's going to lack a mitochondria, Golgi bodies, and peroxisomes. These diplomonads are going to consist and have two equal-sized nuclei and multiple flagella. Giardia is going to be the one that you see here. It infects the small intestine and occasionally the bile ducts in humans and other animals, and can interfere with digestion and absorption of nutrients. So it causes severe diarrhea. A lot of people who hike or go through trails like the Appalachian Trail or the ACT and PCT, things like that, they are going to get this when they drink out of streams if they don't filter their water or don't treat the water before they drink it. Euglenozoa are going to be another type. We have euglenids. Those are going to be photoautotropic unicellular microbes. They're going to contain chloroplast containing light absorbing pigments. They have a paramyelin. That's going to be a complex of polysaccharides that store as food. They have a pellicle. That's a semi-rigid, proteinaceous, helical structure that's going to help maintain the shape of this organism. You also have what's called the kinetoplastids. These are going to contain single large mitochondrion. A kinetoplast is going to be a unique region of DNA that's going to be found within the mitochondrion. So examples of these could be the tryptosoma, and that's going to be the one you see in pink there. It's a causative agent of African sleeping sickness and Chagas disease. It's going to be transmitted by insect vectors like flies and bugs. The next one is Leishmania. This is going to cause a variety of diseases that include Kala Azar and oriental sores. Now we have what we call the alveolates. The alveoli are going to be small membrane-bound cavities beneath the cell's surface, and they have no known purpose that we know of. Three subgroups of these, we're going to have the ciliates. They contain cilia in which they're going to use to move themselves or to move water past their cell surface. They are chemoheterotrophs, which means they're using this to perform organic compounds for both energy and carbon. They contain 2 nuclei, 1 macronucleus and 1 micronucleus, and a paramecium is going to be the well-known pond water ciliate. So that's what you see here in the picture. The next type of alveolite is going to be called a epipicomplexans. This is going to be a chemoautotrophic pathogen of animals. Its intracellular organelles are going to be special complex structures that are going to be found at pieces and infective stages. They are able to penetrate the host. You have the plasmodium that you see here, and that's going to be a slide of red blood cells. It's going to be recognized as the causative agent of malaria, and it is involved with insect vectors like mosquitoes. Now with this, if you have sickle cell disease, then you are not, or if you're a carrier of sickle cell disease, then you are not susceptible to malaria. Our next one is going to be toxoplasma. That's recognized as the causative agent of toxoplasmosis. That's going to give you things like fever, a rash, enlarged lymph nodes, and eye disturbances. It infects all kinds of animals and birds, and it's going to be transmitted from contact with feces or flesh of infected animals. And the last one on here, you see at the bottom, is going to be your cryptosporidium. That's the most common cause of cryptosporidiosis. That causes severe diarrhea. It affects the small intestine and can affect the lungs and the gallbladder. This one's going to be transmitted through animal feces and contaminated water. The dinoflagellates are going to be unicellular photosynthetic microbes, and they're going to make up a large portion of freshwater and marine plankton. They're going to be responsible for things like red tides. Rhizaria is going to be unicellular, and it moves and feeds by means of pseudopodia. So these are going to be movable extensions of their cytoplasm. They also have these little thread-like extensions as well. They reproduce by a way of binary fission. So you have a parental cell that's going to divide into 2 identical daughter cells. The parental cell then disappears with the formation of the offspring. You have foraminifera, and that's going to be a marine amoeba, and that's going to contain porous shells that are going to be composed of calcium carbonate. You also can have a rhodiolaria, and that's going to be a marine amoeba that's going to live part of plankton. So it's going to contain a shell that's composed of silica. Next, we have our amoebozoa. Anytime you see zoa behind it, that's going to mean it's kind of an animal-like. So it contains lobe-shaped pseudopodia. It does not contain shells. And so some of those types here, we have acanthamiba, and that's a free-living soil or water-found amoeba. That is going to be found to cause things like eye infections or keratitis, skin infections, and primary amoebic meningoencephalitis. It has a rapidly fatal infection of the brain or the meninges. If you get this, it could cause extensive hemorrhaging or tissue damage in the brain. The next type that you see here in blue is going to be your entamoeba. That's going to live in a large intestine of humans and other animals, and about 10% of infections are going to cause dysentery. Severe infections will cause things like nausea, cramps, abdominal tenderness, dysentery, and dehydration. Next, we have our plasmodial slime molds. Slime molds are going to lack cell walls and obtain nutrients through phagocytosis. So you'll have 2 different types. You have plasmodial slime molds. These are going to be composed of multinucleated filaments of cytoplasm, and they're going to be found in the forest litter. They phagocytize organic debris and bacteria, and plasmodiums are going to be bodies that contain millions of diploid nuclei. You're going to have these cellular slime molds. These are going to be individuals that are going to be haploid Myex amoeba. So the Myex amoeba is going to be look and act like unicellular amoebas, and they are going to phagocytize things like bacteria, yeast, dung, and decaying vegetation. Next, we're going to go into a different group. This group is the fungi. Their main characteristics are going to be that they are chemoautotrophic, or chemoheterotrophic, sorry. That means that they are going to use chemicals from other things for food. They contain cell walls that are going to be made up of chitin. That's going to be a strong, flexible, nitrogenous polysaccharide. It provides support and protection for the fungi. It also gives the fungi their characteristic shape. They're going to lack chloroplast and chlorophyll, so they cannot photosynthesize. So some fungi significances. Well, they are going to be saprotrophs. Those are decomposers that break down dead, decaying material and release nutrients into soil. They have mycorrhizae. That's associated with about 90% of all vascular plants that are going to aid forest trees and other plants with the uptake of minerals and water. They help to produce antibiotics that help protect plant infections by these bacteria. They're also going to serve as a food source for other organisms, including humans. They're used as food processing, for example, the production of things like cheeses, beer, wine, bread, and things like that. They're going to be a source of antibiotics, so things like penicillin. Penicillin is going to come from penicillium, and that's going to be used to control pathogenic bacteria that's going to be inside of human bodies. Important in research and tools, we use them to study things like metabolism, growth, and development. And about 30% of fungi are going to produce mycosis, or fungal disease of plants, animals, and humans. Now we're going to get into the fungi morphology. So with this, you can have the first thing is going to be called a thallus. If you look at that, the thallus is going to be the vegetative, non-reproductive body of a fungus. So with molds, it's going to be large, long, branched, and tubular. With yeast, it's going to be small, globular, and composed of a single cell. Dimorphic thalli that are going to have characteristics of both mold and yeast thalli, and they're going to be produced in response to environmental conditions like temperature and carbon dioxide. Hyphae are going to be the microscopic thread-like filaments. They can either be septate, which means they're divided into cells by cross walls called septa, or aseptate, means not divided by septa, and they can be multinucleated as well. Mycelium are going to be filamentous masses or a mat of hyphae that are intertwined with each other. You can have two different types. You can have what's called a vegetative. That's where it extends downward into the nutrient material responsible for the nutrient absorption. And you can have the reproductive. That's extending upward into the air, and that's responsible for reproduction of that fungi. Wrong way. Okay, fungi nutrition. So we can have what's called the saprotrophic. That's going to be where it acquires nutrients by absorption from dead organisms. We have hyostoria. That's going to be modified hyphae that are going to allow the fungi to obtain nutrients from living organisms, and they're produced by parasitic fungi most of the time. Most fungi are going to be aerobic, which means that they do require oxygen, though we do have some forms that are going to be anaerobic. So many yeast are going to be facultative anaerobes, and they're going to obtain energy from fermentation. This is going to be found in the digestive system of things like herbivores that are going to help them to assist in plant material catabolism or breaking that plant material down. Some fungi are going to use ionizing radiation, so that's going to be used as an energy source for their metabolism. Now with fungi reproduction, they're going to have two types. They have budding and asexual spore formation. So you have four main types of this. So we have binary fission. That's going to be where that parental cell divides into two identical daughter cells. And from there, the parental cell is then going to disappear once we have the formation of the offspring. And then you have budding. That's a process in which the outgrowth of the parent cell is going to receive a copy of the genetic information. It enlarges, and then it detaches from that parent. You can also have fragmentation. That's going to be where the hyphae are going to break into little pieces or fragments, and each one is going to have the potential to form a new fungus. With the spore formation, we're going to have spores that are going to form at the ends of specialized hyphae, and so this is what you're going to see here in this picture. There's going to be three main categories of spores. You could have sporangiospores. They're going to form inside of a sac called a sporangium. You could have chlamydiospores. They're going to form within a thickened cell wall inside the hyphae. Or you could have cnidiospores, and they're going to be produced at the tips or the sides of the hyphae, but not within a sac. So sexual spore formation has three main types. You have plasmogamy, that's the joining of the cytoplasm. It requires decomposition of the cell walls that are going to separate the hyphae that are involved. You have karyogamy, and that's going to involve the joining of two haploid nuclei. That's going to result in the formation of a diploid cell called a zygote. This involves plus type and minus type haploid nuclei. So that's what you're going to see there with the pluses and minuses. So no sexual dysmorphism of fungi are going to engage in sexual reproduction. They're going to be designated as pluses or minuses in that strain. You have meiosis, which is going to involve the separation of chromosomes and the division of the diploid nucleus into two haploid parts. Zygomycota is going to be a conocytic, multinucleated, saprotrophic mold. The zygosporangia are going to be rough-walled sexual structures. They're going to be formed from the fusion of sexually compatible hyphae, and then you're going to have what's called microsporidia. These are going to be obligate intracellular parasites. So some examples of these would be like Nosemia, Encephala, Platazoon, the microsporidium. These all can cause diseases. Next, we have what's called ascomycota. These are going to be characterized by the formation of haploid ascospores within sacs called asci. The ascocarps are going to be fruiting bodies that contain asci. They reproduce both sexually and asexually, and they include most of the fungi that spoil food, as well as plant pathogens such as agents of Dutch elm disease. They are going to be beneficial in things like baking and brewing. They can be beneficial in research and beneficial as pharmaceuticals. The deuteromycetes are going to be fungi with unknown sexual stages. They're either terrestrial saprobes or pathogenic plants or other fungi and humans. Next, we have the category Basidomycota. These are going to consist of mushrooms and what we call puffballs. The basidocarps are going to be the fruiting bodies, and they consist of tightly woven hyphae that are going to extend into multiple projections called basidia. Basidia is going to be the ends that produce the sexual basidiospores. They're responsible for decomposing cellulose and lignin in dead plants, and they return the nutrients back to the soil. Many are going to produce hallucinatory chemicals or toxins. So the one that you see there to the left is the one they often refer to as the Alice in Wonderland mushroom, or the mushroom that they often talk about with hallucinations. Next, we have our lichens. These are going to be organisms that are composed of fungi living in partnership with photosynthetic microbes. They reproduce by spores. We have Suridia. These are going to be bits of lichen that are going to be dispersed by wind, rain, and small animals to new locations where they can then be established as a new lichen. Lichens usually have three basic shapes. You could see all the way to the right-hand side, we have this fructose shape. That's going to be erect or hanging cylinders. The one that you see in the center is called the crustose shape or crustose shape. That's growth that's going to be oppressed in substrate. And then you have the foliose. That's a leaf-like, so that grows free from the substrate. Food sources for mammals and birds are what usually are the purpose, or what these lichens are used for. And they're going to be used in the production of food, dyes, clothing, perfumes, medicines, as well as they help to monitor air quality. So if the air quality isn't good, then you will start to see the lichens disappearing in the areas that they normally grow. Next, we're moving on to algae. Their main characteristics are that they're eukaryotic and they're phototropic. They carry out oxygenate photosynthesis using chlorophyll. They contain cell walls, which are going to be made of polysaccharides of cellulose or agar. They contain quantities of glass or silica dioxide. They give algae cells their characteristic shape and allow them to live in their hypertonic environments without being damaged. And they don't have stems, leaves, or roots. Algae distribution is going to be mostly aquatic. They're going to live in photic zones where the sunlight can actually penetrate through the water, and so they can actually be found in freshwater, brackish water, or salt water. Algae morphology, they could exist as unicellular organisms, they can exist in colonies, or they can have multicellular bodies called thalli. The thalli are going to be composed of branched filaments or of sheets. For reproduction, they're going to have two types. They have asexual reproduction. In four main types of these, for the asexual, they could have fission. So we have binary fission. That's where the parental cell divides into two identical daughter cells, and it involves mitosis and cytokinesis. Two different types of binary fission. You can have regular binary fission, where it divides in half across the long axis, or you could have what's called longitudinal fission, and that's where it divides in half lengthwise. Next, we have what's called shrinking division. That's a specialized type of fission that results in the formation of two cells of unequal size. Sometimes it's going to produce asexual spores. Fragmentation is going to be where the algae starts to break into little pieces or fragments, and each fragment is going to have the potential of forming a new algae. And then you have spore formation. So spore formation is going to form asexual spores of various types. And so those will float off, land, and then start a new algae formation. Next, we'll have the second type, which is sexual spore formation. And there's going to be three of these. We have what's called plasmogamy. That's the joining of the protoplasm. This is going to require decomposition of cell walls that are going to separate the cells involved. You have karyogamy, and that's going to involve the joining of two haploid nuclei, and that results in the formation of a diploid cell called a zygote. And then you have meiosis. That involves the separation of chromosomes and the division of the diploid nucleus into two haploid parts. Now with algae, it is common to have alternation of generations. This is going to be a method of sexual reproduction in which diploid thalli alternate between haploid thalli. And so you can see that in this diagram here. With the algae classification, the first one we're going to talk about is the Chlorophyta. This is a unicellular or filamentous green algae. It's going to share numerous characteristics with plants. For example, it uses chlorophyll A and chlorophyll B. It will store sugar and starch as food reserves. And many are going to have cell walls that are composed of cellulose. Some are going to have protein cell walls, and others may lack cell walls altogether. Next, we have what's called our Rhodophyta. This is characterized by red accessory pigments that are called phytoerythrin. These are going to contain glycogen as a storage molecule. That's how humans and animals are going to store starch, so as a glycogen molecule. They have cell walls that are composed of agar and carrageenan. They're going to be used as thickening agents in microbial media and food for things like ice cream, syrups, and even in salad dressings. You might have what's called spermatia, and that's going to be a non-motile male gamete for this type. Next, we have our Phaeophyta. This is going to be characterized by brown pigments called xanthophile. So with the xanthophile, it also is going to contain a keratin and chlorophyll in it. Most are going to be marine organisms. They're going to use polysaccharide, laminarin, and oils as food reserves. They're going to have cell walls that are composed of cellulose and alginic acid. That alginic acid is going to be used for a thickener and emulsifier in foods. Next, we have our Chrysophyta. This is going to exist as a unicellular or colonial organism. They contain keratin, which is going to be an orange-colored pigment, and chlorophylls. They use polysaccharides and different types of oils and things as storage products. Some are going to lack cell walls. They're going to have external coverings such as scales and plates on them, and diatoms. Diatoms are going to contain silica cell walls that are composed of frustules. That means they're going to have two halves that fit together. Major components of phytoplankton, and they're going to be major sources of the world's oxygen. Next, we have our water molds. Water molds are going to be numerous and have many characteristics. So they're going to be tubular cristae in the middle of their mitochondria. They have cell walls of cellulose. They're going to have two different kinds of flagella. They have a whip-like and a hairy flagella, and they're going to have diploid bodies. They decompose dead animals and return nutrients back to the environment, and they could actually be a crop pathogen as well. And so with the crop pathogens, things like Phytophthora infestans is going to be a potato famine that was in Ireland during the mid-19th century. What you see on that fish there is a slime mold or a water mold that will start to grow on the fish if they have a damaged coat on their body. So helminths and vectors. Two reasons these multicellular parasites are going to be included in microbiology. One, the eggs and larvae of immature forms are going to be microscopic. And two, many external parasites are going to be vectors, and they involve the transmission of disease-causing agents, including things like bacteria, viruses, protozoa, and other multicellular parasites. Two different categories of parasites could be things like endoparasites, that's endoparasites. Those are found living inside their host. The next is ectoparasites, ECTO. Those are found living outside of their host. So with the helminths, you're going to have the parasitic worms, or the Ascaris. So these are going to be studied because the eggs or larvae are going to be microscopic. They must be distinguished as parasites in microscopic form from other microbes. These also could include things like flatworms and roundworms. So these worms, as you see, can get into adult intestines, and they can fill the intestines. Next, going into our arthropod vectors. These are going to be animals that carry pathogens. They have segmented bodies, hard external skeletons made of chitin, and they'll have jointed legs. Two different types of vectors. You can have a mechanical vector, which means it carries a pathogen, or you can have a biological vector. That means it serves as a host for microorganisms. Two different classes of arthropods will have the arachnids, which include ticks and mites, even chiggers. Ticks are going to be serving as vectors for bacterial, viral, and protozoan disease. They cause things like Lyme's disease, Rocky Mountain spotted fever, relapse fever, tick-borne encephalitis, and even more common, now we have things like alpha-gal. Mites are going to transmit things like rickettsial pox and scrub typhus. Now coming down to the insects, you're going to have things like fleas, lice, flies, mosquitoes, and kissing bugs. Fleas are going to serve as intermediate host for tapeworms. They can also transmit things like the plague that's carried by rat fleas. Lice are going to be parasites that transmit disease. They are vectors in outbreaks of typhus. Flies can be blood-sucking flies which transmit disease such as African sleeping sickness. With mosquitoes, we have the most important arthropod vector. They're going to carry pathogens that cause malaria, yellow fever, and viral encephalitis. And then the kissing bug, which you see last, is going to be important in transmitting what's called Chagas disease. That's going to finish us up for Unit 7. I will see you back for Microbiology's Unit 8.